Welcome to Organelles of the Cell. So before we start up with this chapter, it's important to redefine what an organelle is. What it effectively is a tiny organ-like structure within the cell that carries out a particular function for the cell. Think about your organs. Your organs have unique functions, right? Your brain, your stomach, your liver, your spleen, etc. They all carry out unique functions for your body. And so organelle literally means tiny organs. And so organelles are like organs for the cells. They're going to carry out particular functions that help the cell carry out its overall function. This lecture, it's not really a full chapter, but this lecture is going to look at the various organelles. You're going to want to be able to identify them visually. If you're looking at a diagram or schematic of a cell, and you're going to want to know what they do. So think of this talk like a tour of the cell, basically. One important thing to note, your body's cells are very specialized. So if you were to look at a muscle cell versus a neuron, which would be a cell in nervous tissue, they're not going to look the same. They're going to have different amounts of various organelles, very different structurally depending on their function. And so when we're looking at the cell in this chapter, we're looking at a hypothetical, generalized cell. We're not looking at a cell that has any particular modification for a particular function. We're looking at just a generalized body cell. At least the animal cell. There's also plant cells in this chapter. Speaking of which, here are pictures of the two. You can guess which one's the plant cell, I hope, the one on the right, because of the cell walls. Even though it has a cell wall, plant cells still have the same protective layer that you have in your cells, which is the cell membrane. So it's in both animal and plant cells, and as far as I know, the cells of every living thing on the planet. I'm sure there might be an exception somewhere, but as far as I know, from bacteria all the way up to us, all living cells have the same plasma membrane structure. One thing to note, you'll see it referred to as the plasma membrane and the cell membrane. For our purposes, those two terms are going to be interchangeable. But it's the outer protective layer of the cell. And if you remember back to the lipids section of the macromolecules stuff that we talked about, we mentioned phospholipids and how these make up the outer structure of the cell membrane. So if you remember back, a phospholipid looked like this. It had a hydrophilic head, that means it loves water, hydrophobic tails, which repel water. And so if you remember back, the cell membrane is built from not just one layer of phospholipids, but two layers with the hydrophilic heads of one layer facing inward toward the cytoplasmic fluid, called the cytosol, which is aqueous, it's water-based, and so the hydrophilic heads face in that way, the hydrophobic tails are facing this way. The other half of this bilayer has the hydrophilic heads facing out toward the extracellular matrix, which is also aqueous, water-based, and the hydrophobic tails are facing inward, and so the tails get to face inward, never touch water, and the hydrophilic heads on both sides of this bilayer face in and out toward water-based solutions. So animal cells, this is the only outer layer of protection. In plant cells, you have the cell wall that surrounds this as well. But in both of them, you've got the plasma membrane, and it's made up of this bilayer of phospholipids. Now I want you to look at this very oversimplified picture. You've got the outer membrane made of phospholipids, and you've got this thing right here. You may have recognized it as the nucleus, which means that the DNA is stored inside this purple area. But what is this part surrounding that purple nucleus? That is called the nuclear envelope, and it is a membrane made from the exact same stuff as the outer membrane. You may have recognized this as a very poor illustration of mitochondria. These are some vesicles. This and this and this. The outer membranes of all of those organelles are made up of the same stuff as the outer membrane of the cell. It's made up of the phospholipid bilayer. And outer, right? Outside, outer, one of the prefixes for that is ecto or exo. That's where the word exit comes from. The opposite of that, inside, is the prefix endo. That's where the word enter comes from. But endo means inside, and so any membrane that occurs within the confines of this outer membrane, so any membrane that occurs in here is called endomembrane. And I'm going to show you a little animation of sorts that hopefully will help with this. So in addition to that outer membrane, cells have a large amount of membrane within the outer membrane. And so whenever you hear the word or see the word membrane, I want you to remember that it is made up of that phospholipid bilayer that we just showed. Please remember that. When you hear the word membrane, it's built from this stuff. All of it, whether it's inside the cell's outer membrane or whether it's the outer membrane itself. Watch this animation. That is probably the most generic picture of a cell you could ever get. Let's go ahead and animate it. This is a molecule that we need to have enter the cell. How are we going to do that? Well, how about some of the membrane shift, because it's quite mobile, and engulf that solid particle, molecule, and move it around wherever it needs to go. So notice how this right here is made of the same stuff as this. It's membrane, it's made of the phospholipids. But this creates nice little compartments in which we can do stuff and move stuff and create stuff and whatever else the cell needs to do. And since it's membrane inside of the outer membrane, it's called endomembrane. The reason I'm hammering this term in so much into your brains is because it's going to recur a lot throughout the rest of the semester. So endomembrane, membrane within the membrane, and this kind of gives you a visual of how it works. It creates nice little compartments, right, that separate whatever's happening in here from the cytoplasmic fluid out here, and lets us do various things in those compartments. In fact, there are a number of organelles where structurally all they are is membrane separating what's inside of them from this fluid out here. We're going to come back to this slide later. I want to do this one first. It says, several organelles consist entirely of endomembrane. membrane. This is the nucleus up here. What I'm pointing to is the nuclear envelope, that is membrane. This dark blue is the inside of the rough ER, stands for endoplasmic reticulum. The outer part of it is made up of the same membrane as the outside of the cell. This is called the Golgi body. The orange part inside is separated from the rest of the cytosol or cytoplasmic fluid. This is a lysosome. Pink part is separated from the rest of the fluid, and so it's going to carry out its unique function in there. You get the idea, hopefully. All of the different colors, other than the light blue, which is the cytosol or cytoplasmic fluid, all of the colors other than that are specialized compartments surrounded by endomembrane. That's structurally what they are. Different things happen inside of them, but structurally that's what they all are. So let's start with the cytoplasmic fluid, or cytosol. I'm guessing most of you have heard the word cytoplasm before, but you haven't heard the word cytosol. So cytoplasm technically refers not only to the jelly-like fluid, that's the blue part, inside of the cell, but also all of the other stuff inside of it. In other words, technically, both the fluid and all of the stuff within the cell's outer membrane is considered the cytoplasm. When I learned this, I was taught that just the fluid, the jelly-like fluid, was cytoplasm, but that has since changed. So if you've heard of cytoplasm before, that's maybe what you knew. But we're going to learn it this way. So cytosol is the jelly-like fluid that takes up a good bulk of space within the cell's outer membrane. When you include the cytosol and all of the other organelles that are in with it, pretty much all the contents of the cell, then we're talking cytoplasm. So we've got the outer membrane, you've got hopefully an understanding of how this endomembrane idea works. What's a good place to go then from here? What's a good organelle to start with? I think a good one to start with is where the DNA is kept, which is the nucleus. I've got the nucleus colorized here, the rest of it is gray, in both the animal and plant cell. So what is the nucleus's primary function? The nucleus contains deoxyribonucleic acid. Here's a three-dimensional picture of the nucleus. Ultimately, DNA directs not only this cell's activity, but it directs the creation of proteins, which are used to potentially build other structures and or other cells throughout an organism. And so DNA is the control center of the cell. We mentioned that the nucleus is surrounded by a membrane. It's actually a double membrane called the nuclear envelope. In this picture, you can see these things here. These are pores that regulate the movement of material in and out of the nucleus. One thing that would need to come out of the nucleus would be RNA, because it is going to be what's ultimately read by the cell to build proteins, because the DNA never actually leaves the confines of the nucleus. And so the DNA is here. RNA, among other things, will exit the nucleus. Other molecules may enter. But that's the primary function of the nucleus. It houses the cell's DNA. The DNA never leaves. And again, the nuclear envelope is the double membrane. It regulates materials in and out of the nucleus. The middle part of the nucleus has a particular function beyond DNA storage. Now I want you to pay close attention to the spelling of this, because this trips a lot of people up. So nucleus is the structure as a whole where the DNA is housed. Within the center of the nucleus is this region right here. It's called the nucleolus. And this is going to be where ribosomes get made. What the heck is a ribosome? Well, let's answer that. I want to first make sure you understand what synthesized means. Synthesized means to make. So, this is where ribosomes get built. What the heck is a ribosome? If you look at this picture, and I doubt you're going to be able to see this on my cell phone video, but if you look at the PowerPoint, you can see little dots surrounding the nucleus, and down here on the rough ER, which we'll get to in a moment, there are a lot of them as well. These small dot-like structures are ribosomes. So what do they do? We mentioned that DNA stays within the nucleus. Let's draw a cell. Beautiful cell. Here's our nucleus. DNA does not leave the nucleus. But DNA is the blueprint for building an organism, and many organisms, yourself included, are built principally of proteins. So, DNA doesn't leave the nucleus, but a photocopy of the DNA, a copy of information relating to a particular gene, is transcribed into a type of RNA called messenger RNA. This is like a copy of the original instructions. Think of it like a photocopy of the blueprint for building a house, for instance. That photocopy is carried out to the actual construction site, which is what a ribosome is going to be. So, ribosomes build proteins, which constitute the bulk of you, for instance. So they build you, and they build you based on the instructions that they get from messenger RNA, which is a photocopy of your, not literally, but figuratively, a photocopy of your DNA. So ribosomes build proteins. And ribosomes themselves are built in that center part of the nucleus called the nucleolus. Okay. Now, the little ribosomes are colorized. I doubt you can see that from the cell phone. But I've got arrows pointing to them. You've got some what are called free ribosomes, which are in the cytosol, and then we've got several ribosomes attached up here to this thing called the rough ER. Let's do the ribosome part first before we get to the rough ER. Ribosomes build proteins using instructions from your DNA. So again, DNA is the original blueprint. Messenger RNA takes a copy of that information, takes it out of the nucleus to the construction site, which is the ribosome, where the proteins get built, and those proteins ultimately make up you. So ribosomes have two subunits. You can see those there. Your cells that have that produce a large amount of protein have higher numbers of ribosomes, like we mentioned at the beginning. Cells have different amounts of various organelles depending on their functions. But ultimately, the ribosomes generally work in tandem with the rough ER. So let's go to that one. This blue is the rough endoplasmic reticulum, aka the rough ER. Dotted with ribosomes. In fact, it's called the rough ER because if you were to blow this up and make it three-dimensional and run your fingers against it, you'd feel a lot of bumps of ribosomes. It's very rough in that regard. Remember endomembrane, the structure of it. This is cytosol. This is membrane. And so in this blue area, unique functions are going to happen isolated from the rest of the cytosol in this compartment. And there's various compartments you can see there. So the rough endoplasmic reticulum is made up of endomembrane. It's called rough because it has ribosomes on it. How does this work with the ribosomes? Well, since the ribosomes make proteins, I'm going to guess that the rough ER has some function related to these proteins, and it does. How it works is proteins that get made by the ribosome get pumped into the rough ER. So I'm going to do what will probably be a terrible drawing up here of how that works. So out here is the cytosol. In here is the rough ER. So this is our endomembrane, that blue part right there. That's the endomembrane separating the cytosol from the rough ER. And this would go on and be a lot bigger than this, but you get the idea. The ribosomes are right up against the rough ER, so this is our ribosome. RNA is a single strand of information, so here's our strand of RNA, bringing the information from the DNA. Ribosome gets it, reads the instructions, churns out a nice three-dimensional protein based on the instructions that it got ultimately from your DNA. Now what happens? Well, if you remember the endomembrane here, the endomembrane of the rough ER, it can move around the cell, which means our protein here can go over. It's not going to look exactly the same, so just be aware of that. Pretty close. Our protein will go here. The endomembrane will bud off that and eventually separate completely from the rough ER. Now what we've got is we've got this sac of membrane, a mobile sac of membrane that can move wherever it needs to go, carrying that protein that the ribosome just made. And there's a name for that sac of membrane. Now we're going to go back to this slide. A sac of membrane is called a vesicle, and so this is a transport vesicle. It is transporting our protein from the rough ER off to the next step where it's going to get modified. But vesicles are sacs of membrane. They can fuse with other sacs of membrane. They can also form by going off of a organelle of endomembrane, but don't really worry about this part right now. They transport molecules throughout the cell, into and out of the cell as well, but they transport molecules throughout the cell. That's what I really want you to be aware of right here. And so our transport vesicle is going to carry our protein off to the next step. And here's a three-dimensional picture kind of showing you what I showed. The formation of the vesicle as it buds off of the rough ER. And that purple thing here would be the protein. This is the ribosome. The pink ribbon is the messenger RNA. So our ribosome on the rough ER made a protein. A transport vesicle would have carried that protein across the cytosol down to this organelle here. This organelle is called the Golgi body, and there's a path of travel for our vesicle carrying the protein. The Golgi body, also called the Golgi apparatus, receives vesicles containing proteins that were made by ribosomes in the rough ER. Think of it like the post office or the UPS for the cell. Proteins come into the Golgi body, they're going to be compartmentalized. If there's multiple proteins going to the same destination, they might be united in a particular larger vesicle. Certain sugars might be added as markers onto various proteins. Think of it like a giant shipping warehouse for the cell. Proteins are modified, organized, and ultimately shipped once they pass through the entire Golgi complex. And generally, once they get through this complex, the Golgi body, they're modified to the point that they can now be secreted out of the cell itself. Because often proteins will be created that need to go somewhere else in the organism or in the body. Here's a nice big three-dimensional view of it all. Rough ER with ribosomes that produce proteins based on the instructions of DNA. Proteins travel by vesicles to the Golgi body, where ultimately they travel through it, modified, packaged as necessary. A secretory vessel transports it out of the Golgi body to the end of the cell. This is the outer membrane, where that vesicle can fuse and release the proteins into the extracellular fluid to go wherever they need to go. So that is effectively how proteins get produced within a cell. So the rough ER is called rough because it has the ribosomes. There's a smooth ER as well, which does not have ribosomes. It's made of the same structure, it's endomembrane, but there's no ribosomes attached. Since the purpose of the rough ER involves the protein synthesis, which those ribosomes help with, I'm going to guess that the smooth ER's function is very different from the roughs. And it is. Here's a picture, that same picture of the cell with the smooth ER highlighted. The smooth endoplasmic reticulum is structurally similar to the rough ER, but there are no ribosomes. So they must not be involved in making proteins, and it is not. It's involved in a couple of things. The two things you want to know about the smooth ER is that it makes or synthesizes lipids here, and it helps with detoxification. So if you were to look at a transmission electron microscope image of liver cells and look up close at the contents of those cells, you would see a large amount of smooth ER because the liver is the organ in the body principally involved in detoxification, specifically alcohol detoxification. So we've defined what a vesicle is. Right? This was one transport vesicle that moved the protein around. What if a vesicle is really big? We give it a different name. And that name is vacuoles. So a vacuole is a very large vesicle with a particular function. And we're going to look at one particular type of vacuole. Animal cells have vacuoles as well, but the central vacuole is a unique structure that plants have that helps plants to maintain their rigid structure. So the central vacuole is this large structure here. It's the yellow part. This yellow part is fluid filled, and when it fills with fluid, it helps to excrete pressure on the entirety of the cell, which pushes outward in the cell wall and helps keep the cell rigid and what's called turgid. And part of that is what helps with plants maintaining an upright or rigid structure. So in plants, the central vacuole fills with water. There's also dissolved nutrients in there, but it fills with water, and that helps to maintain the turgid, rigid appearance of the individual cells, which leads to the plant as a whole having the rigid, upright structure. If you stop watering your plants, they might start to wilt. Part of that is because the loss of turgor pressure as a result of the central vacuole not excreting the same pressure from a lack of water. So plants have central vacuoles. You have vacuoles too, but not this kind. One thing you have that most plants do not have is called a lysosome. So one thing you get to do that most plants don't get to do is eat. And when you eat, you eventually have to break down the food that you consume. Lysosomes are one of the final stages of the breakdown process. So you eat food, you chew it, your stomach mechanically and chemically begins digestion, your intestine further digests what is no longer really food at this point, but by the time it gets into your cells, it's usually quite small, but it's still large enough that we need to break it down a bit more. And that's what lysosomes do. Lysosomes are sacs of membrane that contain digestive enzymes. So enzymes are proteins that speed up chemical reactions. In this case, they speed up the breakdown of certain foods. Lysosomes move throughout the cytosol of animal cells, searching for food vacuoles to merge with and ultimately break down the food within. They're also good recyclers. They find old, worn-out organelles, break them down, so your body can reuse the membrane that made them up. Let's look at a visual of how this works. Most plants don't have it. I think carnivorous plants like Venus flytraps would have this, but most don't. So here is what's left of the food that you consumed by the time it's going to enter the cell. Here is the cell's plasma membrane. That membrane is going to form a vacuole, which is, remember, just a large vesicle, which is going to create this little sac of membrane, this compartment containing the food. Digestive enzymes are inside of your lysosome, which is also a sac of enzyme, or a sac of membrane. It's going to ultimately fuse with the food vacuole because they're made from the same stuff. They fuse, the digestive enzymes within are released, and it breaks down what's left of that food item that you had consumed. Speaking of food, I like food. I had pancakes and eggs and orange juice this morning. It was very good. So you consume food to get energy from it. One of the functions, one of the properties of living things was that all living things need to harness energy. And you need specific organelles in your cell to do that. This guy's eating pizza, which sounds really good. I might have pizza for dinner, now that I think about it. The pizza has a fair amount of energy. This guy, as well as you, are incapable. You're incapable of producing your own food. You can't go out into the sun and harness the energy and use that to power your cells. You just don't have the cellular machinery for it. Plants do. They are autotrophs. Remember that term from before? They produce food using the sun's energy. They convert carbon dioxide into sugars. This guy eating the pizza, as well as all other animals, including yourself. You're a heterotroph. You have to consume some other organism to get energy because you don't have the organelle necessary to harvest energy from the sun. So let's start with the autotrophs. What is the organelle that they have that you and I don't, that lets them conduct photosynthesis? The organelle is chloroplast, but the specific molecule that helps to capture sunlight is chlorophyll. It's a green pigment, but it captures sunlight. That's why if you see a part of a plant that is green, that section of the plant is capable of conducting photosynthesis. If it's not green, it can't do it. Because without the green pigment, you can't harness sunlight. Let's do chloroplasts here. Chloroplasts help to conduct photosynthesis, which is the building up of sugar from CO2. So here we go. This is a chemical reaction. Our reactants are what we start with. Carbon dioxide, it is in the air, it is in the atmosphere surrounding all of these plants and other autotrophs. Of course, you need water. Whenever I try and have a garden or grow anything, I usually forget to water it. That's why most of my plants end up dying. So you need water, carbon dioxide, and specifically the equation for photosynthesis is 6 CO2 plus 6 H2O is converted into sugar, C6H12O6, plus oxygen, molecular oxygen, 6 molecules of O2. This is the stuff that plants use some of and then excrete a lot of, and we breathe it in. Carbon dioxide has very little energy. Sugar has a lot of energy. Where does it come from? Energy is not created nor destroyed. Well, it comes in via the sun. You and I might not be able to do photosynthesis, but we can do the reverse equation. If you look, this is exactly the reverse. The reactants of cellular respiration are sugar and oxygen. The products are carbon dioxide and water. You and I do this when we break down food. We consume food, in this case, sugar. We need to breathe oxygen, O2. We produce the waste product, CO2, and water as a result of the breakdown of our food. Once again, energy is not created nor destroyed, so where does the energy that was in the sugar go to? It is ultimately used to power our cells. And how that whole thing works, we have a whole section on later in the semester. But these two chemical reactions are occurring in animals and in plants, and they require specialized organelles to carry them out. Starting with photosynthesis, that process requires, generally, chloroplasts. There are some photosynthetic bacteria that have other methods, but generally you need a chloroplast. Chloroplasts are found only in things that can use the sun to produce their own food. They're green in this picture because of the presence of the pigment chlorophyll. Again, you can't harness sunlight without chlorophyll. If it's not green, it's not doing photosynthesis. Simple as that. Chloroplasts are required to conduct photosynthesis. And again, like we mentioned, energy-poor carbon dioxide is converted into energy-rich sugars. And the energy comes from the sun. So chloroplasts do photosynthesis. What organelle is necessary for cellular respiration? What organelle is needed to break apart food and harness the energy within. That would be the powerhouse of the cell, right? Mitochondria. Just so you're aware, mitochondria is actually a plural word. The singular form is mitochondrion. So if you hear me say that, it's the same thing. That's just the singular. So the mitochondria convert food energy, which is not usable, into energy that is usable. I don't know if we've talked about ATP yet or not, but we'll do it now. So ATP stands for adenosine triphosphate. Okay? Energy has many different forms. Okay? Energy found in the bonds of food cannot be directly used by your cells. It just doesn't work. Think of it like trying to put a quarter into a coin machine that fits dimes. It's just not going to work. Energy stored in ATP that your cells can use. Energy can't be created or destroyed, but it can definitely be converted. And so the mitochondria's job is to take the energy that you've obtained by eating food, for instance, pancakes, which is what I had this morning. My body's job is to take that energy in the pancakes and convert it into ATP energy. And once it's in this form, then my cells can come in, use it, and start to do some work. So it's all about energy converting. The process of breaking down food is called cellular respiration. That was the equation that we looked at. So if plants make their own food, do they still need mitochondria to do this cellular respiration process? And the answer to that is yes, they do. Why do they still need mitochondria if they make their own food? Well, it lies in the purpose of the mitochondria. The purpose is to take food and convert it into ATP. Plants can make all the food they want. You can produce all of the quarters that you want, they still aren't going to fit into a dime-sized coin machine. Only when you convert those quarters into dimes can you start to put those coins in the machine where they belong. And so, likewise, plants can produce their own food with these chloroplasts, but they still need to break that food down by converting it into ATP. And so they have mitochondria as well. So if you think back to the picture that I showed you, this picture, the cytosol is the fluid within the cell. It looks like the organelles will just be floating around in here. There's not a lot of order or structure, seemingly, to this. That would be a false idea, because within the confines of the cell's plasma membrane, you have a whole bunch of protein fibers that collectively are called the cytoskeleton. So cyto, you might run into this word, prefix, more than once. Cyto means cell, and so cytoskeleton means the skeleton of the cell, so to speak. Plant cells may have cell walls, but they still have it too. I doubt you can see the cytoskeleton fibers from the cell phone video. And that's okay, because this is not at all how they actually look. There are a lot of them. They're very thin, but there are many, many cytoskeleton fibers. So what do? They're little protein fibers, large amounts of them, found throughout the cytoplasm of the cell. General purpose of the cytoskeleton: provide structural support and assist in the overall cellular shape and movement, as well as the movement of organelles within and throughout the cell. These guys provide a lot of structure that you really don't see because they're so thin. The time when you can see them best is when the cell divides, and that's a whole other unit in the future as well. The three types of cytoskeleton fibers are microtubules, microfilaments, and intermediate filaments. Microtubules, we're going to talk a lot more about them later. They are involved in what's called mitosis and meiosis, which is cell division. Microtubules, they're hollow, as you might guess by the name, tubule. They're made up of tubulin proteins, and they're really easy to take apart and reassemble and disassemble. Microfilaments are small. They tend to reinforce the cell's plasma membrane, so you have the phospholipid bilayer that makes up the plasma membrane, but you've also got microfilament reinforcement. You can see that they're quite small. The actin subunits that make up microfilaments. And then you have intermediate filaments, which contain fibrous building blocks. And that looks like a very strong, almost cable-like structure. They help to reinforce the cell's shape. I'm not too concerned that you understand a lot of intricacy about the different types of fibers. Be sure that you're aware of what they are, and also be aware of what builds each of them. A term that we mentioned before is tissues. Tissues are cells that are working together to perform a particular function. With animal cells, you don't have cell walls that nicely and neatly hold cells together, so you have to have another method of keeping cells together. And animal cells use proteins to do that. Two particular proteins that are used to fasten cells together are tight junctions and anchoring junctions. Both of these allow for structural integrity so that you do not have gaps between neighboring cells, and that cells are nice and tightly compacted together to form whatever tissue is being produced by their union. So tight junction and anchoring junction allow for structural integrity of neighboring cells. Gap junctions, I kind of jumped the gun here on these terms. So cell junctions allow for the interaction and structural integrity of neighboring cells. Tight and anchoring junctions help keep cells compact together. We said that. They're made of proteins. Gap junctions allow for the transport of molecules between neighboring cells. If you can see, it's not going to zoom in. There we go. Still might not be able to see it on the cell phone, but this protein forms a channel between neighboring cells that allows the transport of molecules. Proteins do a lot of stuff. And so they are, in this case, serving not only to maintain structural integrity in the form of tight and anchoring junctions, but also allow for movement of molecules between neighboring cells. As you mentioned, plant cells, they have cell walls, and so for them, that structural integrity isn't as much of an issue. They have specialized gaps called plasmodesmata, which don't worry about that. But that effectively concludes the tour of the cell. I think that's a pretty good summary of the organelles within the cell and the functions that they carry out.